Alright, so I just wanted to take a moment today and share with you this genetics note that I got from one of my previous class that I made. So I think it sums up the entire unit pretty well, and it's very close to what's usually on the test. So let's start. DNA stands for bone nucleic acid. It is a molecule that stores all the genetic information of all living organisms. It composes repeating units that are called nucleotides, and each of these nucleotides composes of a deoxyribose sugar, which is a phosphate group and one of the four nitrogen nitrogenous bases, and ATCG, and A will always pair with T, C will always pair with G. Basically, this tells us the fundamentals of DNA, which is our first class, I believe. And then the DNA tells us what the cell is going to do. It controls the structure, basically how to do it. DNA replication is a process that occurs before cell division to ensure that each daughter cell receives a complete set of genetic material. During this replication process, the DNA molecule separates and each strand serves as a template of a formation of a new brand. So here's review of the cell cycle, which was, mitosis from grade 9 and 10. So we have interphase. This is when the cell is growing. The cell grows larger and carries out normal growing process. And then the S phase, the DNA is replicated. In the G2 phase, the cell continues to grow but prepares for mitosis. And then mitosis is then separated into four phases. Prophase, which is the nuclear membrane and nucleolus disappearing. This is the first phase, and then you will start to see them start to split apart. Sorry, not split apart, but you will start to see that the membrane is gone, and now we have the spindle fibers, which is going to be the things that the chromosomes is going to attached to. And then in the metaphase, the chromosomes line up in the middle, and they attach themselves to the spindle fibers. While the anaphase, they're pulled to the opposite sides. The centromeres, which is the thing that attach the strand onto the spindle fibers, it will eventually break, and this will create single-stranded chromosomes. Well, chromatids. And finally, it's the telophase. This is when the chromosomes decompose to become chromatins, and the cell membrane folds inward while the nuclear membrane is back. Now, there's something that I highlighted in red is that important that mitosis only takes place in somatic cells. This is because, well, it's going to produce perfect, well, daughter cells. It's going to be completely the same. Everything is going to be completely genetically identical. So cytokinesis is the last process, and finally we have after cytokinesis we've successfully created two genetically identical diploid daughter cells. It's very perfect, and there's very little amount of chance that you're actually going to get any mistakes. So just like mitosis occurs in somatic cells, we have meiosis that occurs in reproductive cells. So this is where gametes are produced, sperms and eggs. It reduces chromosome by half, ensuring genetic diversity, and it involves two rounds of division, which is meiosis 1 and meiosis 2. So it divides the chromosome by half. It's because usually, well, not usually, always you need a dad and you will need a mom. And when the guys combine together, you're going to get a perfect individual. So unlike, well, mitosis, we start off chromosomes from, both parents. So let's say I have a dad, I have a mom, and then the sperm and the egg meet, and we're gonna have, chromosomes. And then together they're called tetrads. They're gonna link up right here in prophase 1. They're gonna pair up. For example, for the same trait, they're gonna get paired up. Let's say if I have dad's hair color and mom's hair color, and then they're gonna be on the same trait, so they're on the same chromosome, and these two chromosomes will pair up. But okay, I was just an example because a chromosome is too big, way too big to just say the hair color. It's gonna say many other stuff. So the chromosomes describing the same trait is gonna line up together, and they're gonna form tetrads. So basically four chromatins. So we have, well, two chromosomes basically. So they pair up, and then all of these happen which is basically metaphase, anaphase, telophase. Everything is the same thing. It's the same process as mitosis, except you're starting with more and you're ending with more, obviously. But one big difference that occurs in meiosis that do not occur in mitosis is crossing over. So this is where the stage of the tetrads is formed. But when we split them apart, we can't just split it perfectly. Because if we split them perfectly, there's going to be no genetic diversity. And what does that mean? That means you and your sister and your brother, you guys are going to be completely identical except for gender. So there's a little bit of crossing over, which means that your eye color might be a little or a lot darker than your brother's, and your brother's hair color might be a lot darker than yours. And then after that, we have meiosis too. This is completely similar to mitosis, but except for that we create more. So basically, since we had two chromosomes to form tetrads, we break down the tetrads to form two cells that has two chromosomes, and then obviously meiosis too. We break those again, and then we will create four genetically unique haploid gamete cells. They're all genetically unique, and it's different because all of them is supposed to be different, or else humans will be extinct, I guess. And then we have oogenesis and spermatogenesis. Basically, that's creating sperms and eggs. So oogenesis occurs in females. We produce only one usable eggs and polar body. So basically, when we are onto meiosis too, and we create these four genetically unique haploid gamete cells, we do create four, but there's only one of them is a usable one. This is because the unequal distribution of cytoplasm. And meiosis 1 for this production of egg usually happens before birth. For females, meiosis 2 does not begin until the sperm fertilizes the egg and finishes meiosis. Now, spermatogenesis loses cytoplasm every step it goes through. That's why sperm cells are really small. But it uses that energy that it produces to actually develop a flagellum, which is basically many people they say that uh, the sperms, they look like they look like something that can swim, and obviously it is true. They can swim. I've heard many examples of people saying sperm looks like tadpole, which is pretty accurate because it kind of does. It has that same tail as a tadpole, and it can swim, and that's how it reaches the egg. Anyways, these energy will be stored in the mitochondria in the neck. At the end of meiosis, the spermatogenesis will produce four viable sperm cells. So basically, woman only creates one egg, one usable egg, and I spent a whole lesson talking about it because it was some sexual health thing that I had to address as well. So I spent a whole class addressing about how how people get pregnant and how females eggs work and how sperm works and blah blah blah. Now let's move on to mutations and genetic disorders. So a mutation is any change in a DNA sequence that leads to some sort of variation. So mutation can obviously occur naturally, or it can be induced by environmental factors. For example, radiation and nuclear active stuff usually cause some sort of reaction, and it causes people to have itches, skin rashes, and such. So type of mutations include point mutation, which is a change in a single nucleotide, or a frameshift mutation, which means an entire nucleotide is inserted, and then the protein sequence is completely screwed, and now you're just a whole different person. So genetic disorders are mutations that cause genetic disorders. Obviously, this can be inherited or occur spontaneously. We talk about Down syndrome, Edwards syndrome, Patty syndrome, Klinefelter syndrome, Turner syndrome, and the only one that you actually need to remember is Down. And I expect you to remember Edwards syndrome, but it's okay. So the chance of getting these syndromes greatly increase as the pregnant lady gets older. So if you know if there was a pregnant woman coming into the hospital and she's at 20 years old, we usually wouldn't be giving her a screening and be like, oh, your children is safe from Down syndrome. But if we have a woman that's coming in at 45 years old, we'll be like, okay, you know what, we got to do a lot of searches to make sure that your kid is healthy. So this trisomy and monosomy, this is okay. So I haven't talked about this. There's not much in the notes, so I will talk about it. So this is when you have 3 chromosomes instead of 1, which is not so good. So in the 18th set of chromosomes, so human, we have we have sets of chromosomes. In the 18th, there's 3 instead of regular 2. And obviously, Klinefelter is in the gender, it's usually XX or XY. However, it's an XXY, which is not good. So these occurs when an individual has 3 copies of a particular chromosome instead of 2. This is because of non-disjunction, an error during meiosis, where the chromosomes or sister chromatids failed to separate properly. So this actually happens. Now we talk about a little bit of Mendel and traditional genetics. So Mendel was a monk. He planted pea plants, and for the most time, the pea plants came out yellow and green. And then he found out all of these funny things. He breeded them, and they found 2 laws. Well, 3, but I didn't write that. Law of segregation is that there's always a 2 version of a trait passed through yourself. So basically, you have one from dad, one from mom, and unfortunately, you can only pass one to your offspring because he needs one from dad, one from mom, and you're either the dad or you're the mom, so you only get to pass one. Law of independent assortment says that the chance of getting a particular trait does not affect the chance of getting other traits. However, this is not true for some special cases, but he summed it up good enough. So in here we did monohybrid and dihybrid crosses, and I 100% on both. Well, I believe I gave you guys the practice quiz. That was a good 100, yeah. And then codominance and incomplete multiple alleles. So monohybrid and dihybrid crosses. If you have any questions, you can go back to the videos before. I believe you shouldn't have any questions, but if you have, comment down below. So codominance, incomplete dominance, and multiple alleles is a little different. It's the special cases. So codominance, when you have both traits are fully expressed. For example, a black pig with white dots, or a white pig with black dots. So incomplete dominance is none of them is dominant, so it really results in a blend. So basically, when you have an orange cat and a white cat, it blend out to be a calico cat, and it looks cute. Multiple alleles is that more than 2 alleles control 4 genes. So usually we have big A, small a, regular big 2 big A's, homozygous small a, or something. But then we have a B, B, C, C, D, D, E, E, F, F, G, G, whatever. All of these are for height. However, they're all separate. So if it was that genes, he will either pass. So when you pass on these, it's usually the dad will, let's say he passed on A, B, C, D, E, F. So we treat them as separately. We treat this as 1. So mom passes small a or dad passes small a, and then we treat B as 1. Mom passes small b, dad passes big B, whatever. And then blood types. Oh, this was stupidly confusing. I agree with this person right here. I'm not gonna drop the name, but I agree with him. It's very stupidly confusing. So there are 4 types of blood: A, B, B, and O. So, however, the genotypes is different because we can have homozygous big A, we can have A mixed with a heterozygote with a little O right there, heterozygote. We have homozygous B and B with O, and then we have AB, and then we have II. Only I, so I is kind of recessive. So there's also an RH factor, and so RH factor positive is dominant to negative. So if a person is typo negative, he can only be IIRH minus RH minus. So when you really memorize this, just remember this: everyone can receive blood from type O minus, and but nobody except for O minus can give blood to O minus. And then type AB can take every single type of blood. Remember that type AB minus cannot actually take blood from type AB plus, which is unfortunate. And that was that. So polygenic inheritance is a trait is controlled by multiple genes. This was taught earlier. And then heterozygote advantages talks about how carriers have certain that has some heterozygotes. They usually have a survival benefit, and I gave an example here. Lethal genes is that these genes are fatal, and then when they're present in homozygous form, like a homozygous recessive, or like if you say right here, Manx cat, the talonness is dominant, but but these dominant trait, when you have a homozygous of it, they will die before birth. So, and when we do a cross, we actually do not count the dead individuals because we just don't count. And then pedigree analysis, we did a lot of it. I gave a lot of practices, and you should have found more of it. We talked about regular pedigree, some advanced stuff, and then that's pretty much it. And then the last lesson was reproductive strategies, where we talk about how humans mix up many things, and we do biotechnologies, all of these. Yeah, you don't exactly need to understand this, well, remember it, but you do need to have a pretty good foundation of us trying to manipulate as much of DNA as we could to potentially help us fix many diseases and virus. And this part, I think the hardest emphasize that we always do is that biotechnology is a stupidly fast growing field, because biology might have existed for a good amount of time, but biology hasn't. It was a very recent science, and it heavily depends on chemistry and physics to actually look at stuff with microscopes. So if you are interested, remember to look into this part. And that's pretty much all the notes. I will be taking you all through a practice test a couple, well, maybe today or tomorrow. But yep, that's all I gotta say today.